So I'm going to talk about near-infrared spectroscopy, the how and the why. I haven't written a recent review on muscle NIRS, but those of you in the field, I commend you to look at Tom Barstow's recent review in 2019. It's an excellent review about both the methods and the terminology and how you should save your data in the results, how you should present your results. So unfortunately, it's not open access, but either me or Thomas have the paper. If you need to get one, you can send us an email. So what I'm going to talk about today is what does NIRS measure? How does it measure it? In particular, similarities and differences between machines. Then the break for the questions on the technology. Briefly talk about quantitative physiology measurements with NIRS, and then some of the practical uses of NIRS, in particular my own studies in elite sport. And finally, some new ideas I've thrown out about potentially new ways of analyzing and using NIRS. So what does it measure? Well, I'm sure you're very familiar that NIRS reports indirectly on the oxygen delivery that we pick up in the lungs and the consumption inside the mitochondria, in this case, inside a muscle cell. Obviously, nearly all the oxygen in our blood is carried on the red blood cell protein hemoglobin, about 95% or more. And there's a conformational change involved with the hemoglobin protein binding and releasing oxygen. And that conformation change, just as well for us, has a color change associated with it, where it goes from a sort of bright red color in oxy to a sort of wonderful kind of mauve color, crimson color in deoxy. And that's the spectra for the pure protein, but also in blood, you'll see the same color change. So we don't use visible light because that is such a strong absorbance, no light gets through the tissue, but near-infrared light between 700 and 900 nanometers can pass through tissue, as you can see here. Obviously, when it passes through tissue, it must interact with the tissue in some way. So although there's not super strong absorbance, there's enough absorbance of the light by chromophores so we can report information about the tissue. And the dominant chromophores in biology that are in the near-infrared region from 700 to 900 are oxyhemoglobin, deoxyhemoglobin, and the mitochondrial protein cytochrome C oxidase, which is a bit trickier to measure with near-infrared. I can answer questions of people interested in the end. There's an additional chromophore in muscle called myoglobin. There's a protein sitting inside the muscle cell, very similar color to hemoglobin. And the contribution of myoglobin to the signal is, depending on who you speak to and which paper you look at, anywhere up to 50% of the signal. So it's actually pretty important in terms of interpreting the data. However, most people ignore it in a sense, and I'm going to kind of ignore it in this talk. So you need to assume I'm talking about myoglobin as well when I talk about hemoglobin. I didn't always ignore it. Honestly, I went back to look at my sort of slides for this talk and look at my ideas in the past. And 10 years ago, 2009, we got together a really good team to actually put together what we call the Big Muscle Grant. We were going to look at myoglobin and solve the problem with near-infrared and of myoglobin measurements and how useful they would be. We had a physicist leading the project. We had sports scientists. We had physiologists. We had clinicians. We had NMR spectroscopists, because you can measure myoglobin with NMR spectroscopy, engineers on the team. So it was about £2 or £3 million grant. We thought we'd solve the problem, 5-year grant, didn't even get beyond the first stage of review. So obviously not sexy enough for the funding bodies, unfortunately. And then the engineers and physicists all went off to do sexier studies on the brain. They left the muscle. And I went off to do sexier studies, I guess, on elite athletes. So that's where the myoglobin story kind of ended for us, unfortunately. It's kind of a shame, but couldn't get the funding to follow it up. So if we're talking about measurement, how the NIRS measurement works, there's interesting and fairly obvious links to how pulse oximeters work. The pulse oximeters, I'm sure you're aware, and we're all aware because of this time of COVID, how important they are, that measures arterial hemoglobin saturation, tells you how well the lungs oxygenate blood. It links into the arterial pulse signal, so it only measures arterial blood, whereas the infrared spectroscopy goes deeper in the tissue and measures a combination probably of arterial, the smaller capillaries and the small veins. Exactly what proportion is not clear. It's generally assumed about a third, probably because that's a convenient thing to write. The problem is not only is we don't know exactly what this combination is, but it might change during potentially during more extreme protocols. So that is an issue you need to be aware of, where the signal comes from. So how do we convert the light signal to the chromophore concentrations that we're interested in to report on oxygenation? This is the absorbance spectrum in the near-infrared of oxy and deoxyhemoglobin. And if you get a conversion of oxy to deoxyhemoglobin, and you measure at, say, 760 nanometers, you'll see an increase in absorbance. If you measure at 880 here, for example, you'll see a decrease in absorbance. And if you measured on the isospecific point here, you'd see no change in absorbance. If you saw a change in oxy and deoxy, they both changed, that would give you a signal here as well, because both these chromophores would increase and absorb light. So essentially, we've got two unknowns, so we need at least two wavelengths to determine the different concentrations. And indeed, this is a picture from 2016. All muscle neuron devices use a number of wavelengths. Some use more than two, but they all use a minimum of two. And for most of my talk, I'm going to be focusing on portable devices. And really, at the moment, there's only two out there of any meaningful use, as far as I can tell. Obviously, the Portamon and the Moxie monitor. And the Portamon uses two, and the Moxie uses 4 wavelengths. So I'll come back to that later on. So what about similarities or differences between these different machines? Because they give you what looks like it might be the same number, but is it really the same number? How do they work? How do you compare your data you're getting with somebody else's data, which might use a different machine, even though it's measuring infrared? So how do they work? Why are there so many machines? Are they indeed different? First question everyone asks is, where does the light go? And the first slide everyone shows is a slide like this, which is kind of a working conceptual hallucination of where the light goes, but it's sort of backed by optical modeling. So it goes somewhere like this, in this kind of banana shape, into the tissue, through the skin, through the epidermis, through the fat layer, into the muscle, and comes out again. And as it does so, the oxygen is consumed and the oxy is converted to deoxy. And depending on the source detector separation, you get different depth penetration. So depth penetration will not just depend on the nature of the layers, but also on your source detector separation. So here's a sort of a 4 centimeter source detector separation, which is typical for the Artinis Portamon, and you expect to get about 2 centimeters of light penetration. And this is about the most simple maths I'm going to show, probably the only maths I'm going to show, is that depth of penetration therefore is half the source detector separation. But that's only a rule of thumb. There might be ways of getting around that, and I'll show you some tricks people use to try and get better access in the depth, the deeper tissues, to avoid some of the surface layers they're not really interested in getting information from. So how do you measure a concentration with light? I'm only briefly going to mention this because Thierry, in his talk earlier on, described it in a bit more detail. Essentially, you use the modified Beer-Lambert law to convert a optical density change to a concentration change. The assumption in the very basic, simplest continuous wave near-infrared spectroscopy is that there's no change in light scattering. So the only change you detect is a change due to a change in oxy or deoxyhemoglobin, or myoglobin, of course. And if you assume that, you get a change, an absolute change, but in concentration terms. So deoxyhemoglobin would be micromolar, but it's a change from a baseline. And you can have an oxyhemoglobin change, and you can have a total change, oxy plus deoxy, potentially related to blood flow. And you can have an oxygenation change, oxy minus deoxy. And all machines that I've shown you could produce these outputs. They don't all, so you won't be able to get these outputs of a Moxie, for example, even though in principle, the data is there to do that. Now, I guess because they're so in love with pulse oximetry, because it's a wonderful technique, everyone thinks it's really important to get an absolute hemoglobin saturation, but that's been the gold standard, the idea, the dream for NIRS technology for a long time. And therefore, a number of people have tried to think about ways of doing that. And I'm going to show you two different methods of doing that in portable near-infrared spectroscopy. The first is called the SRS, or spatially resolved spectroscopy. And that's illustrated by the Artinis Portamon. Second doesn't really have a well-characterized acronym, but I'm going to call it spatial resolution spectroscopy, or SR. And the illustration of that device is a MOXIE. First, with the Artinis device that I'm sure you're all familiar with, that takes advantage of the fact that light scattering beyond a certain distance is homogeneous. And by that I mean, here is a smartphone with a camera sending a white light into a bottle of scatterer, which is probably a dilute suspension of milk. And what you can see is that other light's coming in from a single source, the amount of light coming out at all the different directions, and it's coming out because of light scattering on the milk molecules, it's the same intensity. So if you're a big enough distance away from the light source, the scattering is the same. So the trick then is to have a light detector, a light source here, and a detector with three sensors very close together. And the scattering then will be the same between these. So if you look at the slope of the amount of light absorbed between these three distances, the scattering will be the same. So for example, according to the maths of light transport, there should be sort of very small separation between these three detectors and a long distance away from the source. But in general, this works. Here's a 3, 3.5, and 4 centimeter. First device to use this was Hamamatsu near-infrared spectrometer, the first commercial device. But you're probably familiar with the Portamon, and that actually uses 3 transmitters and 1 receiver. But the maths is the same. You still get the same equivalent data coming out. So what do you get in the SRS method? Well, you still get your hemoglobin concentration change, or your by oxy and deoxy, but it's now multiplied by a scattering constant, but it's the same scattering. So that constant involved the scattering is the same, and therefore you get an equation like this: absolute oxy, absolute deoxy times this scattering constant. But because it's the same, you can then, if you do a saturation, if you divide the oxy by the oxy plus deoxy, these K's scattering terms cancel out, and you get a oxygen saturation measurement. And this is the way these devices measure things. They're sometimes called different things: tissue oxygenation index, tissue oxygenation saturation. They're all essentially the same thing done by that method. Of course, you still get a number if you add these two terms, the K times the scattering times the oxy and the scattering times the deoxy, but that you can't get rid of this K term then. So this is the sort of device producing the total hemoglobin, and it's therefore the total heme, because it will include myoglobin. It's sort of, I guess, semi-arbitrary units. People tend not to use it, though I think it's potentially quite interesting because it's a depth-separated, a depth-resolved value for total hemoglobin. It's actually quite useful, so I think it should be probably looked at a bit more. So now what I call spatial resolution spectroscopy. How's that different? Well, now we have the light detectors much more separated from the source. So we now have almost equal distances between the source and the first detector, and the first detector and the second detector. So for example, you might have a 1.25 and then 2.5 here. And the idea of these devices, and there are very many different ones of them, is to cancel out a superficial layer. So in head, you're trying to cancel out the skull, and the muscle, you're generally trying to cancel out the skin, but especially the fat layers. So what you do with these devices, you make a measurement at, let's say, detector A1 and detector A2, and then you have some algorithm to simply look at the difference between the light at these two places and use that to calculate a tissue oxidation saturation or a muscle oxidation saturation, whatever you want to call it. The issue here is that all devices use different algorithms, and some of them are commercial, but they're certainly not the same. So while every machine that uses SRS will broadly use the same algorithm, it's not true that every other machine that gives you an oxygen saturation number and is set up like this kind of setup will give you the same number or will even use the same technology. So there's 3 devices. There's 2 devices predominantly used in brain at the moment, the foresight and the INVOS. And here's the MOXIE used in muscle. These all use different algorithms, so they're not comparable in terms of how they make their measurement. But they all give you an absolute number. So what we're going to do, how do we compare these absolute numbers? And I'm going to compare 2 different devices in muscle, some work we did a couple of years ago, comparing the Portamon and SRS method with a MOXIE, which is a spatial resolution method. And to illustrate, this shows you, here's the Portamon with the 3, 3.5, and 4, and here's the MOXIE with the 1.5 and the 2.5. So they're different source detectors. Now, they're therefore going to use different algorithms. Now, there's a slight advantage of the Artinis because they've got a longer 4 centimeter difference. So they're going to get better depth separation anyway because of that. But then you pay an awful lot more money for the Artinis than the MOXIE. So you sort of pay your money, you take your choice, really. Okay, this now shows you the difference between the 2 devices in an exercise test. And this is on measuring on the arm a 30% maximal voluntary contraction or 50%. And what you see is both devices show a fall in saturation, in the contraction, and the bigger the contraction, the bigger the saturation. So they're going to correlate. If you compared MVC versus the device saturation, they'll correlate, but the absolute number won't be the same. And you can see that obviously from looking at this slide. Interestingly, in this particular case, the MOXIE is giving you a bigger apparent resolution and a bigger signal for what you're seeing. You can say, well, that's real or not, but it certainly gives you a difference. So they correlate. Which one is right? Who knows, actually? I mean, there are, I wouldn't say thousands, but there are certainly tens of papers comparing one non-gold standard near-infrared spectroscopy technique that gives you a number with another one. And because they're all designed to measure saturation, they all kind of correlate. But the actual numbers are always different, and it's not clear which is best, or even where they're met. And they've got different source detector separations. They're measuring in different parts of the tissue. Anyway, so even the absolute number shouldn't necessarily be the same. But we did explore this difference, particularly between the MOXIE and the Portamon, a bit more to see what could be causing the differences in this particular case. And what we looked at was, you saw, we measured just basic resting vastus lateralis on the thigh muscle oxygenation. There was very little difference, well, no difference between males and the 2 devices, but the MOXIE had a much higher saturation significantly in females. And that rings bells, because when you're comparing males and females in a difference, the first thing you generally think about is, in NIRS spectroscopy, is surface fat measurements. Because the fat layer is heterogeneous between subjects and certainly between sexes, and females in general have a larger surface adipose tissue layer. And that's shown by this slide here, where we took our resting data, measured the skin fold measurements of the fat level in the vastus lateralis, and the Portamon was rather insensitive to a change. There was a significant change with the skin fold, but it was rather insensitive to changes. These are in different subjects, but the MOXIE was more sensitive to changes. So it could be, one explanation is that the MOXIE absolute number is rather sensitive to the fat device, fat lower. And I think when you look at the actual, recently there was a nice paper published by Roger, who designed the MOXIE and developed by Andre. And what they indeed showed was that the device, looking at skin fold thickness versus the MOXIE saturation, it's now in exercise, so not at rest like I did, in exercise. And there's a small change, but above a certain skin fold thickness, you start to see rather large changes in the saturation number. And this is fairly significant in terms of your subjects, because this sort of level here, where they're starting to see a rather large difference, is basically for the normal, relatively healthy females. So there are issues with the device in measurement in females, and certainly females that didn't have low adipose layers might be problematic. In comparing absolute numbers, the difference changes might still be fine, but the absolute numbers might be more problematic. So why do we get an influence from the fat layer? Well, we're comparing here a surface fat smaller and a larger surface fat layer here. The light will still penetrate tissue layers, but the proportion of the signal you get from the muscle will depend on the fat layer thickness. So clearly we get a bigger depth of light penetration with a smaller surface fat layer. And so with a high fat layer, less muscle volume is interrogated by the light, and the intensity and depth of the detected light is underestimated. So there's an intrinsic problem. So it's always better to have a larger source detector separation if you can anyway, independent of what your algorithm is. So how have people got around and tried to correct for that? Well, there's a number of ways. First, I'm going to describe methods intrinsic to the individual machine or the algorithm the machine uses. So this is a really interesting data from Joan Conway, showing that in fact you could use near-infrared light itself to measure the fat and then correct for it. So this shows you the 930 absorbance band due to fat, and there's also a 970 absorbance band due to water. So you can ratio these two and get an estimation of the fat content in tissue using the same device that is making your in vivo measurement, and then apply some kind of correction. And that's what was done by Professor Kolberais-Mateus, Koblenz, and a number of really interesting papers where he used all the gold standard ways of measuring adipose tissue thickness, ultrasound, MR imaging, and then calipers, and used them optically as well. Here's showing his correlation between the optical measurement and, in this case, the ultrasound thickness, and used that to develop a Monte Carlo-based model of what the thickness of the fat was, and using that then to correct his tissue oxygenation measurement. So this is directly all intrinsic to the single device. It's a home-built device. You can't buy it, I'm afraid. To show you the kind of differences that gave you, this is a cyclist, I think it's a male cyclist actually, exercising with increased power, and this is without the model, the fat model correction. You do get a desaturation and then you get a recovery, but you get a bigger dynamic range and a bigger desaturation when you correct for the adipose tissue layer using the 970, 930 peak actually. So that's a really interesting way of going about it, but it's technically quite difficult to do, and you need to have extra wavelengths in your device. The spatial resolution methods also try and do a fat correction. And they do it by an algorithm where they say the short distance is mostly going to be biased towards the surface, towards the skin and the fat, and the longer one will be more muscle. So we'll bias our signal, create an algorithm, and use that to give you more useful information, more muscle-biased signal. I can't go through the whole detail of this, but there's a nice link here to description of how the MOXIE device uses the Monte Carlo two-layer model to try and correct for the fat contribution. Therefore, at least get some way around this issue where they've got a very small source detector separation, but they still want to detect as much light from muscle as they can. Interestingly, I was preparing this talk, I was looking at this MOXIE device, and I looked at the Artinis website. I thought, well, this exactly looks like a Portalight Mini. So in fact, you have the same device you can buy from Artinis, even though you have the same algorithm. And then I thought that is not really an original thought. You could combine these spatial resolution and SRS methods to kind of get the best of both worlds, to correct for the fat layer with this smaller short distance, and then still use the SRS to get your depth penetration improved and your absolute numbers. So it's not a completely original idea, but he's kind of talking about combining a Porta Mini and a Portalight together, a few extra source detector separations, probably only actually one extra distance, really. Now, if you're lucky enough to own an Octomon, you can get a short separation channel. In principle, that might help you get a better NIRS trend signal from the doxine, the deoxine, correct for the fat. But I think it's be really nice to get that integrated and added to the more general portable devices. So there's another way of doing this correction, and that relies on the user inputting the information rather than the machine measuring it, or the machine trying to correct for it even without measuring it. you give the device the information. So you measure the fat by ultrasound or MRI or even just using calipers, and then input that into the machine. And as we all know, models are only as good as the data you input. So you think actually a model would be better for having the input from the individual subject. And there was one device that tried to do that. That was designed by Aston. Unfortunately, it doesn't exist anymore. And in that device, you actually put the device, you measured the skin fold thickness with a caliper, put the device on the subject and typed in the thickness you measured, and they corrected the algorithm by some modeling method. So this is my version of that kind of device. Really, it was because I looked at some data I had from male and female biathletes who were doing incremental running tests. And the females were really not desaturating anywhere near as enough as the males, and they were really fit, elite athletes. So it was quite surprising. So I measured the fat layers, applied a Monte Carlo 2-layer model to it, and I actually brought the data closer together. So this bigger separation without the fat correction came closer together. So that was a promising start, but it was still only a start. There's more work to do. And it's important to note and not be too scared or worried about fat, like it'll all mess up all your data. So it will make a difference, especially when you get larger adipose tissue levels, to the absolute number. But it doesn't affect the overall trend in an individual if you've got a big enough source to get the separation. So even with what my correction does here, each individual is showing the same trend. The fat doesn't change, the fat correction doesn't change the trend, the desaturation, it just changes the absolute numbers. So as long as you're measuring the fat contribution, and as long as the fat contribution doesn't dramatically change, obviously if it's the same individual, unless you're doing a 6-month training program, it shouldn't change very much. And then within that individual that you could apply the correction. So you don't need to apply correction, you just need to measure it and be sure it hasn't changed. So don't get too worried about it, but it's just interesting to note there are ways of getting a better absolute number. Second method I want to talk about is methods that are independent of these instrument manufacturers and these algorithms. So what can we do ourselves as physiologists or biochemists or just users? And these methods are not just about correcting for differences in fat layers, they're also just correcting for any other region, reducing the variability in your measurements. So both variability between machines and between subjects. And the primary one I'm going to discuss is something that's already been briefly mentioned in some of the conversations and questions earlier in a previous webinar, and that's what we call the physiological calibration. And as I say, I think this helps with fat correction, in principle normalizing it to the individual person, but it also helps to standardize tests between and within subjects. Now, I used to be not a fan of this. It was what was done in near-infrared spectroscopy before they had these kind of ways of making an absolute measurement. You created, essentially what you do is create your own 0 and 100%. And I thought, well, the machine's already doing it. We should let the physics do it and not do it ourselves. But I'm becoming a bit more of a fan, and I'll tell you why in a moment. So anyway, how do these methods work? Well, what you do is obviously you've got a number that your manufacturer is giving you, and you then do an arterial occlusion. So you stop the blood going in or out of the tissue. Oxygen will be consumed, and the idea is you'll get to a 0 number. You then release the cuff, oxygen flows back into the tissue, there's an increase in blood flow, you get a hyperemic response, and you get a maximum number. And you then set this to 0 and this to 100%. You're now ignoring the number on the machine other than telling you whether it's really where you are on this scale. You've created your own independent scale, and you use that scale for all your measurements. So if next time you come and the person you're trying to compare people with 2 slightly different fat contents, this might be a better way of doing that. I've got a couple of theoretical problems with that. First of all, is the 0 really 0? And what I mean by that is, I'll show you an example here, is that if you do an arterial occlusion, are you sure you've really got to 0? And this shows you some data in calf muscle where you can see, well, are you 0? It's still trickling down, maybe some blood squeezing in. You're not really sure you're at 0. Unless you do exercise. So that's hard work for people. I mean, an athlete can do it, but I can tell you, doing exercise when you've got an arterial occlusion on is not easy, but you could do it. And then you can be sure, or much surer, you've got to 0. But some tissues you're measuring on, like here's measuring on a tendon where the oxygen consumption is really slow. So you're never really going to get to zero here in any meaningful time. And then what about the reperfusion? So are you really 100%? Well, in one sense, theoretically, you can't be 100%. If 100% means all the hemoglobin, all the myoglobin has got oxygen on it, you can't be that way unless there's no oxygen consumption in the tissue, because you are measuring a combination of not just arteries, you're also measuring the veins and the capillaries. And however fast your blood is flowing, there must be some extraction, and therefore that 100% can't be a real 100%. It will be dependent on some other factors depending on that individual, how much their blood is flowing, what their oxygen consumption is. And this illustrates that point I'm making there, is that here's an arterial occlusion I've done at the end of exercise, and to get myself a 0-100% range. Here's the actual activity here, and if I do the end of the activity, the number's above 100%. So at the end of the exercise activity, the device measures, on this scale here, greater than 100%. So the scale's a bit arbitrary. I have an issue with that, but I still think there's value to it in certainly comparing between different tests you're doing, and certainly between different machines. So going back to my Moxie Portamon, I said the Portamon, the Moxie and the measurements you were making had a larger dynamic range. And this is illustrated here by, I think we're doing an arm cranking study. And this shows you that, yes, they're both desaturating, but they're desaturating differently. But you then did an arterial occlusion at the end of the study. And then you do calibrated measure. And you see with the calibrated measurement, now these two devices are identical. So if like the difference between the two devices, you've corrected by normalizing them to their own physiological differences between arterial and hyperemic response. So I think there's definitely a place for physiological calibration, and I'm becoming gradually more of a fan of it, I have to say. And at that point, I think I've talked long enough on my own, and have anyone got any questions? Hi, Thierry.
Yes, hi, thank you for the talk so far. It's been really interesting. We have a few questions coming in, one from Victor Ochoa. I will allow him to speak. It might be that you already subtly, unintentionally answered this question on slide 63, but...
slide 63, right?
Able to talk right now.
Yeah.
He's still muted, Victor. Could you ask your question, maybe? Still muted. Might be that he doesn't want to talk. I can ask the question. Oh, there we go.
He's unmuted.
Hello.
Hi.
Nice talk, Professor Christopher. I have a question. How do you portray a new generation of NIRS devices, and which will be the challenges?
Next generation. I feel I'm not an instrument manufacturer, so I know what's technically feasible. I do think, as I was talking about, I think we should be certainly having a better correction for these surface tissues and modeling and and people in muscle. I mean, obviously in the brain, because functional near-infrared spectroscopy in the brain is such a big thing, everyone's measuring multiple source detector separations and multi-site measurements. We should be doing that in the muscle. So that will be, I think, the difference. In principle, you can do that, but because it's not cheap, and the problem is the devices, and the people working on the brain have more money, so they can buy more complicated devices. But I certainly think having a shorter source separation to correct for the fact's a good idea, and I certainly would encourage manufacturers to go along with that. But I encourage researchers to get the funding to get something like the Optimon, where they can make multiple measurements at multiple sites, and in principle get topographic mapping. So there's no reason why you can't get similar information in the muscle. Obviously, it's functional data of a different kind in the brain, but that should be possible. There's nothing wrong with that. And in a sense, the advantage of muscle, once you've solved the adipose tissue problem, is that you get a whole lot more lighted muscle. So you want a big advantage. So you can get bigger source separations in muscle because the signal's much, much bigger. So I think I'll leave the instrument manufacturers to say about big technology differences, but certainly that would be possibility, and better portability. I think we should be able to get that sorted as well, and that's pretty good now. I mean, I was surprised how people athletes were happy with the Portamon, but I'm not sure the average person that's already a a quite bulky thing. And then the big question that stops usability is, of course, price. So you have to have a price point that makes money, and that means the market's got to be bigger. So think of the technology in a mobile phone. I mean, a mobile phone is a kind of cheap near-infrared device. In principle, you can get a oxygen saturation number up by putting your thumb on a flash point on a mobile phone. But the technology in a mobile phone is way more advanced in a sense than the technology in a near-infrared device. But it has £1 billion market, so there's people working on it. So I think it's a balance. But I think in terms of getting more metal and muscle, in terms of getting more site measurements, and I'll show in my second half how it's really useful, not just within a single muscle group to get more measurements, but to measure on different muscles at the same time is really important. So I'm not sure if that answers your question, but that's probably what I would like to see happening.
Yeah, thank you.
You're welcome.
Okay. A few more personal questions from Dr. Reed. She didn't put them in the Q&A box, but she said she had a few, so I will unmute her microphone now, and I think she's able to talk. Dr. Reed, are you there?
Can you hear me now?
I can, yeah.
Okay, great. Thank you for a very interesting presentation. I really enjoyed it. I have some questions about one of the first comments you made was about myoglobin and how 50% of the signal might be from myoglobin. I'm looking at the NIRS device from a clinical perspective. So I often work with people who have chronic respiratory disease. So I was wondering, how important is it to ignore that myoglobin signal in different types of exercise or in different clientele? And I realize you work at the other end of the spectrum in that you often work with elite athletes.
Oh, I have worked with patient groups as well. It's not important to ignore it. You mustn't ignore it, and you should report. You should always say your signal is a combination of the different devices. You can't ignore it because unless you're using there is a method that a group in Seattle has got where you can potentially discriminate myoglobin and hemoglobin. Actually, I should have reported to Victor's question. That's what the machine manufacturers should do. But that needs a broadband system. So I think so. You can't ignore it because the signal's there. You have to, obviously, if you're seeing a total heme change, that's almost certainly due to hemoglobin because the myoglobin won't change unless the light path is changing. But you can't ignore it. It's there. You need to put it that it could be from myoglobin. If you try to interpret your data, of course, the signals myoglobin is measured tissue PO2. Essentially intracellular PO2, and hemoglobin is measured vascular PO2. And they're not the same, but in disease, they clearly might not be the same. So we need someone to properly be sure, because the 10% to 50% range, one is animal data, they said 10%, but then some of the human data said 50%. So we need that better characterized. That might need biopsies. But certainly, I think you can't ignore it. It's there, and you have to think about if that's an issue. But going back to the instrument manufacturer question, and it really was related back to what I wanted to do 10 years ago, a broadband system where you measured all the wavelengths of light, and you could certainly use a super continuum system, so you could do lasers. It could be able to separate myoglobin and hemoglobin, but it's tricky, but it's doable. And I think I do spit like trying to measure cytochrome oxidase. It's the equivalent challenge actually to measure the myoglobin and hemoglobin, but it is feasible. So I think that would be really useful and might well be important clinically.
Okay, thank you. I have a second question, but I can wait if there's others that have questions.
Yeah, I suggest to continue with the talk for now and do the rest of the questions at the end of the talk.
Okay.
So thank you for the question.
Okay, thank you. As I say, happy to be emailed about any questions down the line. More than happy to. Shall I carry on?
Yeah, sure. Go for it.
And most of my talks, I'm starting to accelerate through the talk probably now. Hopefully there are references on all the talks. So if you need to query, the papers should be present on that talk, so you can go back and look at the video and pick up the papers. Okay. So I'm very quickly going to go through the quantitative physiology measurements with NIRS, and that's because previous speakers have mentioned it. So in Thierry's own webinar, he talked about measurements of muscle blood flow and muscle oxygen consumption. And you make those measurements, the blood flow by this is Mirel's data, Rorschach Mirel's data, via a venous occlusion, and use an arterial occlusion to get an oxygen consumption measurement. Trickier to do during exercise, of course, but this is just about possible in some exercises, but this isn't easily. So, but you get at rest, venous occlusion and arterial occlusions give you oxygen consumption and blood flow. I should actually talk about devices. There is I should just quickly go back to what Victor's question again, is that there is a DSC device that can measure flow and would be really good to combine that with near-infrared spectroscopy measurement. So in principle, there's a near-infrared way of measuring flow. It's just expensive. So it won't be anywhere near like, you know, £5,000, but that's probably going to come in the future and get cheaper. So that's probably the new way of measuring flow. I'm now going to talk about oxidative capacity, but again, very briefly, this is mitochondrial oxidative capacity, because Alfred Nimmer-Richter yesterday actually gave a really great webinar going through that in some detail. But essentially what you're doing is looking at recovery rates after exercise. And as we nearly all exercise studies show, something that drops to saturation, looking at the recovery rates is really important. And in principle, this just shows you the best way of doing this is that as you get the recovery, you've done the exercise, as you recover the oxygenation, the oxygen goes up, the oxygen goes down. At each point, you do arterial occlusions, and that gives you muscle oxygen consumption methods. And you can measure the return of muscle oxygen consumption to baseline, and that mirrors the rate of phosphocreatine recovery, which you get from NMR spectroscopy. And that has been well known to be a valuation of the skeletal muscle mitochondrial capacity. Now, this is tricky to do. I mean, you can persuade people to do it, but you don't want to do it for every single study. You don't want to be doing arterial occlusions at the end of each exercise test. And it's therefore quite nice that in general, the reoxygenation rates that you see after exercise are pretty similar to what you see from actually making the oxygen consumption measurement. So you should, that's in principle a sort of poor man's way of doing this measurement. The PCR measurement you make, which is linked to the oxygen consumption measurement, correlates that, which correlates with the NIRS recovery, direct recovery. They all correlate with total mitochondrial ATP synthesis rates and total mitochondrial respiratory train content, amount of mitochondria. So it's a bit like one correlation leads to another correlation, leads to another correlation, but in principle, measuring those recovery rates can give you information, albeit indirectly, about mitochondrial content. And that's useful in both health and training, and of course, disease. And there was more on that in yesterday's talk. So finally, I'm going to go over some sports data. I'm going to give, I'm not going to be comprehensive. I'm going to focus on my own work and essentially on my own work with elite athletes. And I'm going to give you highlights of my work and point to similarities or some themes that have arisen and that I think are useful and in the future in thinking about this. So I'm going to start with winter sports. That's where I did my PhD. Niagara Falls in the winter. There's no problem with social distancing. Niagara Falls in the winter. Very cold. No one goes there. So sports studies, I've worked with elite athletes. In particular, I've worked with short track and long track speed skaters and also biathletes. Summer sports, nice bit of social distancing here between the water skier and the boat. I've worked with elite rugby players, elite hockey, and with synchronized swimmers. So using these sports as examples, I'm going to talk about the advantages and the benefits of multi-site measurements. And this probes the biomechanics of the task and can inform the coach and potentially assist with new training ideas. And we've got data, useful data from the speed skaters, from the biathletes, and also from especially from the synchronized swimmers. So the speed skaters I worked with was both the Team GB track team and the Dutch Olympic team as well. And they were completely happy. They loved having the portable device on their legs, in fact, much more than wearing the gas analyzers on their faces. They didn't like that, take that off. But they were happy with the portamons. And there was no data dropout in all the studies, which was great. And no one fell over and damaged the device or their legs, which was good. So nice set of studies I've done in this area. I'm going to just tell you a little bit about the fact of one particular study. And this was comparing the right and the left quadriceps muscles during short track speed skating during a fast time trial. And this is a slide that I feel like made its third showing in the Artinis webinars. This is a short track speed skater doing a time trial. I think this is a 500-meter time trial. When they go off, their desaturation drops in both legs, but with time, it recovers in the left leg, but not in the right leg. And that's because when they go around the corners, they hang on the right leg, and the right leg is compressed. You can see here, there's a compression in the right leg. And they never really recover. So their right leg does an awful lot more work than their left leg. And in fact, not all the female athletes are able to hang for the whole of their event. So it's a real challenge. It's a really difficult thing to do this. And it's very difficult because you're giving yourself these occlusions, and that carries on throughout the race. And the men don't hang all the time. So having noticed this, it was informed some of their changing, and they did change their training. At least they told us they did to strengthen the right leg in training. So make sure they had strong right legs. And also they were interested in developing using nutritional aids such as nitrite or beetroot juice in the form of beetroot juice to increase blood flow. So there was, I wouldn't say it was only NIRS that did this, but it becomes part of the overall coach package. They were using the NIRS information as part of their overall package to inform how to deliver their protocols and training and preparation to the athletes. So this is biathlete now, which is shooting and skiing, where we don't get much winter in the UK. So we ended up doing this roller skiing instead. This is a UK elite biathlete. And what we did here, we measured on the vastus lateralis, on the leg and on the triceps and the arm. And this is just one example. We measured it through a whole range of different protocols, but I'm just illustrating in one point the difference between two different athletes. So this is going downhill. So they're doing no work now. And normally going downhill, it's a chance for them to recover. So recover their saturation, and then they go and have to do lots of hard work because it's a very aerobic intensive sport, skiing. So this shows you athlete B behaving nicely because he's resaturating both his arm and his leg going downhill, preparing for the future. But subject A is presumably tensing up. So he's going downhill and he's still desaturating. So if the coach could see that, these weren't real time, but in principle, they could see that in real time. The coach could have given advice at that point. So this is a sort of, if you like, a case study. It's in the sort of information you could get and delivered in real time. And again, it's about making multi-site measurements. You get lots of extra information making multi-site measurements. So finally going to look at multi-site measurements in swimming. And the first study I'm going to talk about is work we did with swimmers and triathletes when they were swimming. This is a swimmer, and we're asking a swimmer, this is a club level swimmer, to swim as fast as they can. This is now proper ecological. Then they're swimming in a swimming pool with a waterproof device, sprint as fast as they can for 200 meters and then recover. I'm measuring on the vastus and on the latissimus dorsi. So essentially measuring in the leg, and this muscle helps drive the arm movement. So in swimming. So this is what you might expect from an athlete, a swimmer. They desaturate in both their arm and their legs. They try to go as fast as they can. So when swimmers are sprinting, they use both their arms and their legs. If you compare this with a triathlete, in the triathlete, what you do is, of course, you swim, and immediately after your swim, you go on a bike ride. And immediately after a bike ride, you go on a run. So they're desperate when they're swimming to conserve their legs because next two exercises are very leg dependent. So they just don't kick their legs when they're swimming. They certainly don't kick their legs when they're swimming in the actual triathlete, but also when they're training. So if you actually tell a triathlete to swim as fast as they can, they really just don't use their legs very much. So here's the desaturation in the arm, but the legs don't desaturate much at all. So in one sense, this is obvious data because you can see they're not kicking their legs. But but it also tells you what you can see there, you could pick up with the spectroscopy. And it's again telling you the differences between multi-site measurements. And of even greater interest and even more complex was work we did with elite synchronized swimmers. And that was a consultancy role for NHK, a Japanese TV company, and we were helped by Artinis in doing this work. And we made muscle and brain measurements underwater. And the question, I guess the research question, as much as you can ask a research question in a TV program, was, did the brain desaturate in underwater exercise? We're pretty sure the muscle would. But synchronized swimming is a hugely difficult sport. It's very demanding, having to do all this exercise whilst underwater when you can't breathe. So the question is, how do they protect, or do they even protect the brain saturation? And it's a problem. This shows you, I think it's the Beijing Olympics, this is a competitor collapsing after their routine. So they're on the edge with breathing and brain oxygenation, potentially, or we think they might be. So we were asked to do this study, and then eventually a TV documentary was made out of it. Is my student making the measurement on putting the portum on the vastus lateralis, the portum on there, and the portalite on the head, which you can't quite see. So, and I was amazed. They were happy to have the portum on while doing, I mean, synchronized swimming routines are really complicated leg kicks going on. And they did it throughout exactly the same routine because, of course, they do exactly the same routine all the time. And they did it with the device on, and we got really good data, both from the leg and from the head. So it's very impressive, and really a sort of big shout out both to Ben for doing it and to Artinis for the device and to the athletes themselves. So just to remind you, or to show you, we're working with elite athletes. We worked with the Russian, Japanese and Spanish teams, essentially, apart from the odd North American contribution and French, I guess. These are the medal winners in the Olympics. These are the teams to beat. And the Russians especially always get the gold medal. So I'm not going to show you the videos. I did so in a previous talk here, but what I really urge you all to do, if you're remotely interested in the applications of NIRS, is to Google Russia's Golden Mermaids on YouTube. And there should still be, hopefully, if it's not been taken down, a video both in Japanese, if you can speak Japanese, but also an English language version of the whole documentary. And it focuses on why these two Russian athletes, actually one in particular, are so good at doing synchronized swimming, and they stay underwater for much longer. How do they do that? And it's kind of a great detective story, which is why I'm not going to give you a spoiler, but a lot of the answers you get are from making near-infrared spectroscopy measurements on the athletes at multiple sites during and you can map the measurements they do with the video into what's going on in the muscle oxygenation. And I won't do it justice by showing a brief video. I don't want to spoil it for you. I really urge you to go watch the video. It's great. So we had to try and get a publication because we're scientists out of that. So I'm not going to give you a big spoiler, but we did manage to get something published. And broadly speaking, as you would expect, of course, when they're kicking their legs underwater, the muscle desaturates. And the beauty, of course, of synchronized swimmers is that you have your own control. These are two swimmers doing exactly the same thing. So this is subject 3, subject 4. So you can see the similarities. And if there's a difference, you've got the video and you know they're almost doing the same thing. So in principle, you get really interesting information if you track the video to the athlete. But in terms of publication and research question, the basic thing we were able to show was that, yes, during a synchronized swimming routine, on average, the muscle desaturates, but the brain in general doesn't desaturate. So the brain appeared to be protected. So that's a rather simple, rather obvious answer, but it was good to show it. And again, it's the multiple site measurements that made the difference. So I'm finally going to end now on the second aspect of sports studies with elite athletes, and that's informing our training studies by exploring either the extent of the desaturation or recovery rates. So here's an example of I think this is a bike exercise test, going flat out on a bike, kind of a Wingate test. So you show desaturate, you can look at how far they desaturate and also how fast they recover. Or if you've got longer time at the end of the test, you can do a proper recovery half-time measurement. In effect, a mitochondrial capacity measurement. We've got data here from rugby players, from hockey, and from swimmers. So I'm going to start with professional rugby union players, and that was work. Here's my student, Ben Jones, when he was a professional rugby player. And this is Ben attempting to get social distance between himself and this guy, and that social distance is going to turn into 0 in a split second's time when he flattens Ben, I'm afraid. So, but the great thing is Ben went from being in front of the camera as an England Rugby Union player to being behind the computer doing work with me on elite rugby players. So it's great to see Ben doing that kind of work and making that transition. It's great to work with an elite athlete if you can, who's also got a good brain. It's brilliant. So this is the work we did with these elite academy rugby union players, doing a bike test. And what we're showing this shows the deoxyhemoglobin change. In each test, you got desaturation and a recovery, a desaturation and a recovery. And we took 8 players, and what the rugby club were interested in was their pre-season training effective? So we measured them before they started their pre-season training, and then when they were just starting their rugby playing. And what we saw was, yes, that when they were in the season, they had a significantly increased deoxygenation extent. So they looked like they were consuming more oxygen, extracting more oxygen after their pre-season training, which was helpful at the level of the muscle, not just at the level of their, doing their VO2 max tests or whatever. So that was training adaptations in rugby players. What about in swimmers? And now we're working with adolescent club level swimmers. Now we're not doing a bike test, we're actually getting the swimmers to swim in the pool. This is Ben getting up at 5 o'clock in the morning, not me. Ben did it, getting up at 5 o'clock in the morning to go to the swimmers in the swim club, make the NIRS measurements. And what they were doing was 200-meter swim, recovery, 200-meter swim, recovery. And we measured the extent of the saturation change and also the recovery rates, both before and after a training. I think it was a 8-week training program. And what we saw, probably the most interesting result, was this recovery rate. The change, we saw the percent difference in this recovery rates was correlated. The quicker that recovery rate was, the better the improvement in the swim time. So they all improved their swim time, but those who had the faster recovery rate appeared to increase the swim time more. So again, suggesting that this could be useful information about what's going on at the level of the muscle relating to their performance. Finally, and probably the most interesting thing in terms of training that I've done was work I did with the Team GB field hockey players. And they came to us because they knew we were NIRS experts, but they were also interested in changing their training before the Olympics. They were concerned they were getting fatigued at the end of events, so they wanted to up their training capacity, up their aerobic capacity, but they didn't want to do impact work because they were worried about injuries before the Olympics. So they wanted us to trial a bike training program. So we came in, trialed their bike training program, their interval cycling training program, and at the same time made the near-infrared measurements on the leg to see, yes, was there a change in their sprint times or whatever, but also was there a change in their muscle, at the level of the muscle. And what we showed was that post training program we introduced, or the coaches obviously introduced, they had an increased desaturation. So these are cycling, again, these are repeated, this is really hard work, repeated wing gait tests. So it's 1, 2, 3, 4, 5 in a row. After 1 or 2, I normally throw up, but here's five in a row by these women, very fit. And then we measured both the recovery times, the recovery rates, and the extent of the desaturation. And after the training program, they desaturated more during the intense exercise, but they recovered quicker. And so that was a combination of validating their training program they used, and they certainly kept that training program, partly because of what we did, but also obviously we're only one component of a large team, prior to 2016 Olympics. And I'm sure you all remember the 2016 Olympics gold medal game. And the Team GB were on the ropes. They were the last 10 minutes of the match. They were up against the wall. They just about managed to hold out for a 1-1 draw, even though the other team were probably a better team. And then when it came to the penalty shootout, Team GB won the gold medal. And the unfortunate team they beat, of course, you can tell by this orange here, was the Dutch team. So this is a great example of Dutch technology. The Portamon assisting the British team to beat the Dutch team. So perhaps best not to boast about that, Artinis, but we liked it. So I want to just make one other point about the hockey data, and that is, if you want to use the technology as a coach, they always say they might be happy in, saying, well, we're validating your group training program. But what about, can I use it on an individual athlete? And for that, you need to make sure, really, it seems to me, that every individual shows the same training effect. It's no good if the group data shows a significant difference, if the athlete you want to train isn't showing a change. And obviously in individual sports, this is even more important. So we were fortunate in this particular study, and it's not true for all our studies, but this particular one, every single one of the 6 girls who all ended up in the team, actually, that we tested, all showed the same trends, significant differences. But it's really a plea here for everyone to publish their individual as well as their group data, especially in elite athletes when you haven't got very many and it's easy to do. And usually when I'm reviewing NIRS papers for publication, I always say, where's the individual data? Even if it's only supplementary information, it's really good to see that, because if we're going to ever take this technology and use it in the real world as a real world monitor, like you would use a pulse oximeter, it's no good having an average pulse oximeter. That's got to work for that individual. So you've got to work for the individual. You've got to show the same training effect for all individuals. Let us know if you did or not. Publish all the data. Okay, so finally, just a few new analysis ideas. First one is something I did a long time ago, looking at, that when you do cycling with NIRS, you get these rhythmic changes, and that's because the blood, these oxy and deoxy changes and total hemoglobin changes. And that's because the blood is being pumped in and out of the muscle blood pump, as you're turning the wheel. So you might think, oh, that's just noise, but it's not noise, it's real physiology. We were able, with this guy Tiziani Binzoni, really, to develop a method where we could look at, as the blood was being pushed in and pushed out, how much oxygen was being consumed with each cycle, and therefore get a measurement of oxygen consumption, not by doing an occlusion, but just dynamically during the normal exercise, which is kind of really where you want to go. You want a real-time oxygen consumption measurement. You don't want to have to do an arterial occlusion and intervene. So that was in principle really good. The problem was, it turns out the method was quite tricky to use. And when we did it, it only seemed to work. We lumped all the group data together. You needed to use 3 wavelengths to correct for scattering. So it's probably not the ultimate answer, but it was an interesting idea about how you could, in principle, get dynamic information. Another possible thing that we've done with the brain that we might want to move into the into the muscle, is to use a more metabolic modeling or systems modeling approach to aid our data interpretation. This is what we did on a brain circulation and metabolism model to help us interpret NIRS data in the brain from hemoglobin, and actually from cytochrome oxidase as well. And what that gave us, we would put a whole range of systemic and cerebral inputs into the data, and a model then would predict outputs that you couldn't measure. So, for example, the model would give us a predicted brain oxygen consumption, even though we weren't measuring brain oxygen consumption directly. And it would be quite interesting to see if we could use a similar model to aid our NIRS data interpretation in muscle. So, haven't done that yet, would be an interesting thing to do in the future. And finally, I guess a plea for help, or what I would have done if I was in the bar. So this is where the problem with a virtual is that I can't go to the bar with you and ask you questions. You have to ask me questions. I can't ask you questions. So I would say, here's all my training studies. I've done them in swimming, sprint swimming, and repeated Wingate tests on a bike. I get certain things with training that I see the same in my studies, but I don't know whether other people do. I'd like to compare data. So what I mean by that is this shows you either a swimming sprint here, or hockey, and I've lost my pointer, because it's a green pointer. Here's the hockey, here's the swimming, here's the rugby. Each time you exercise, and this is, remember, cycling flat out or swimming flat out, you drop your oxyhemoglobin, increase your deoxyhemoglobin, but you drop the total. There's some vasoconstriction because you're constricting muscle. The hemoglobin concentration drops. And that's true for all 3 studies. So all 3 studies show a drop in saturation, all 3 sports, hockey, swimming, and rugby. they're all measuring on the bastus, show a drop in oxygenation, and however you measure the oxygenation, and also a drop in total hemoglobin. But if you look at the effect post-training, and what you do now is you post minus pre, so you look at the differences post-training and subtract the differences pre-training. So it's a subtraction, if you like, or a subtraction. What's the effect of the training? The effect of the training appears to be, in all three cases, very little effect on this hemoglobin drop, the volume drop, the concentration drop. But there is definitely effect in oxygenation. So I look at this data and I thought in my head, this looks a bit like an arterial occlusion. So there's no change in blood volume, but there is a change in oxygenation. Therefore, oxygen consumption has changed. So in principle, I'd like to see maybe what's going on is we're really just picking up an oxygen consumption change, even by this simple method. But I don't know if that's true. That's what training effect is. I don't know when that training effect actually occurs. So it'd be good to see and compare to other people. I'm going to end on this final complete. First of all, that's what I want to do in the bar. But obviously, you can email me afterwards if you've got good data, and we can talk about it. So the second thing is, my final plea is to publish all your data. Ideally, all the raw data, but as much data as you can cut, even if it's only supplementary information. I say that because this is some work by Martin Buchholz. He did a whole range of great training programs, really good NIRS data he's produced. And this is him showing in an exercise test the fall in oxy and deoxyhemoglobin, the trend monitors, but also a saturation change. So he's using the Portamon, or this is probably the Portamon actually, to its full capacity. But then when he reports his training data, he only showed TSI. So he only showed the saturation change. He didn't show the trend monitor changes. So I can't compare it to the totality of my data. So I've emailed him and he said he doesn't have this data anymore. So it's kind of a plea. Please let you publish all your data if at all possible so other people can compare. Even if they publish it, this lets you put it as supplementary information, get it out there so we can look at the data, because these studies, especially on elite athletes, they're kind of gold dust. You don't get many of them. They're hard to do. We should make sure we curate the data properly. So I'd finally just like to thank and acknowledge people who work with me, Chris McManus in the Moxie work and Ben Jones. They're both talented PhD students of mine now. They're both lecturers in sports science at the University of Essex, doing great. Catherine did all the winter sports work, and she was a great student. She went on to the dark side, not really the dark side. She went to work in sports. So she went away from academic life and she worked helping Team GB in the, she worked for UK Sport for the Rio Olympics, and then she worked in the Football Association. She now works with the English Football Association. So that's her role now. And obviously I'd thank all the athletes and coaches involved in the studies and all the various funders over the years who funded my muscle work. Thank you very much.